Hi there, welcome to AP Bio Fun with Dr. D. This is part 2 of enzymes, and I'm going to reiterate again, enzymes are reusable. Remember this, enzymes are reusable. So let's talk about how environmental factors affect enzyme activity. Remember that in order for the enzymes to function, this substrate must bind to the active site. And in this model here, you will see how this is the shape of the active site. Substrate kind of matches that active site. Once the substrate binds, the active site changes its shape slightly as the substrate binds. This is called induced fit. Products are produced, products leave the active site of the enzyme, and the active site of the enzyme is restored to its original shape. Once at the active site, substrate molecules are bound to the enzyme surface in the right orientation to facilitate reaction. This will lower the activation energy. The substrate binding usually involves hydrogen bonds, ionic bonds, or both. And substrate binding is readily reversible. For this reason, the environmental condition in which an enzyme is placed will affect its ability to function. So, which amino acids do you think are involved in the active site? In the last lecture, I gave them to you as names, but here I'm going to show them to you as formulas, where the R groups are highlighted. Aspartic acid and glutamic acid are what? Look at the side chains, the R groups. What kind of bonds can they form? Versus lysine, arginine, and histidine. What kind of bonds can these amino acids form? Pause the video and think about it by looking at the side chains, the R groups, which are highlighted either in magenta or dark blue here. Hopefully you are able to figure out that these amino acids can form ionic bonds with the substrate because they are charged. Okay. Now, unfortunately, there's only few amino acids that can form ionic bonds. So vitamins to the rescue. Some enzymes contain, actually many enzymes contain non-protein cofactors. They help bind to the substrate, and they're called coenzymes. Coenzymes are made from vitamins. That's why vitamins are essential in our diet, because they help produce these coenzymes, which are essential to the function of so many of our enzymes. Without them, our enzymes cannot function. For instance, FADH, which we're going to encounter later, is produced from vitamin B2. NADH and NADPH are produced from niacin. Both of these are vitamins which are an essential part of our diet. In addition to coenzymes, which are a small non-protein organic molecules, some enzymes are actually using metal ions, such as iron, copper, zinc, magnesium, manganese. Here are some examples. Now, these metal ions, because they're ions, they're charged, help form hydrogen bonds and ionic bonds with the substrate. So they bridge the active site of the enzyme with the substrate. And think, why do supplements, bottle of vitamins often contain these as well? Because again, they're essential part of many, many enzymes in our body. So many enzymes in our body require these metal ions to function. So how do we measure enzyme activity? Well, in order to answer that question, you need to ask yourself, what do enzymes do? What do they do? Well, they speed up chemical reactions. So how do we measure the rate of a chemical reaction? We measure the rate of the chemical reaction and we infer from it how active an enzyme is. But how do we measure the rate of a chemical reaction? We measure the amount of product which is produced over time. So the rate of a chemical reaction, which is catalyzed by an enzyme, is affected by various factors, including the concentration of the enzyme, the concentration of the substrate or substrates, if it's more than one, temperature, pH, pressure, concentration of other solutes. All kinds of factors in the environment can affect the rate of a chemical reaction. But we're going to be talking first about the concentration of the enzyme. So remember when we played the enzyme game, and we got more keys to open the 10 doors. What happened? As we got more and more keys, the rate at which we open 10 doors increased. Same thing happens in chemical reactions. As we add more enzyme, the rate increases up until a certain point where the rate just levels off. Now in cells, however, there's usually a lot more substrate in enzyme. So what we observe is like a straight line. So what else affects the rate of a chemical reaction? Well, the concentration of substrate. So remember when we added more doors in our enzyme game. As we added more doors, the rate increased. But why is the curve flattening? So think, what needs to happen for the enzyme to work on the substrate? The enzyme needs to bind to the substrate. So if you have only a certain amount of molecules of enzyme, if every enzyme molecule is bound to a substrate, it doesn't matter how many more substrates you add, the reaction rate is not going to change because the enzyme needs to bind to the substrate, convert it to the product, kick out the product before it can bind to another substrate molecule. So remember what happened when we added more doors in our enzyme game. So as you increase the substrate molecules, as you have a lot more substrate molecules than enzyme molecules, every enzyme molecule is bound by a substrate at any given time. And we say this is saturation of the enzyme. The enzyme is saturated with substrate. Every enzyme molecule is bound to a substrate. And this is the maximum velocity, the maximum reaction rate of this particular reaction. Depending on the chemical reaction and the type of enzyme, that Vmax, that maximum velocity differs. And it really only depends on the type of chemical reaction and the type of enzyme. So when the enzyme is saturated, when every enzyme is bound by a substrate molecule, we have Vmax, we have maximum rate, reaction rate. All right, what else is affects reaction rates? Well, we have temperature, we have pH, we have pressure. Now this again should be a review for you because you've studied this extensively last year in bio, but let's review briefly. In this graph, I've covered up some of the labels. You see on the y-axis the reaction rate, on the x-axis is temperature. And what you see is a positive slope, like the reaction rate increases as we increase temperature. Why does the reaction rate increase as we increase temperature? What is the reason? We talked about it in the first lecture. As we increase temperature, we increase the kinetic energy of the molecules. That means more molecules will have energy equal to or greater than the activation barrier, the activation energy. So temperature increases reaction rate because at higher temperatures, particles move faster, they have greater kinetic energy, and the frequency of collision is higher. Okay, now this is the second half of the graph. Here I'm showing you the first half of the graph. Here I'm going to show you the second part of the graph. Why is it we reach a certain temperature and past this temperature, now the graph has a negative slope? Now the reaction rate starts rapidly to decrease. Why does the rate decrease after a certain temperature? Tell me, why? And again, you should be familiar with this because high temperature will cause denaturation of the protein, partial or complete unfolding of the protein, tertiary, quaternary structures, even secondary structures will be disrupted. The shape of the active site will change. The enzyme will no longer be able to bind to or bind as efficiently to its substrate, and the reaction rate will rapidly decrease. What about pH? pH has the same effect as temperature. It will disrupt the 3D shape of the active site. It will disrupt. Temperature disrupts hydrogen bonds. pH will disrupt hydrogen bonds. pH will disrupt ionic bonds because you have either too much hydrogen ion or too little hydrogen ion, and you disrupt the balance between all of these interactions, the hydrogen bonds and the ionic bonds that hold together the 3D shape of the protein. The moment the shape of the protein changes, even slightly, the shape of the active site can no longer match the shape of the substrate. Enzyme cannot bind substrate. Enzyme cannot bind as efficiently the substrate. No binding of substrate to enzyme means no catalysis can happen and the reaction rates are plummeting. I want you to quickly look at these curves. On the upper graph, you will see reaction rates as a function of temperature. Lower graphs are reaction rates as a function of pH. Now, these are two different enzymes in both cases. The first graph, you see one enzyme with an optimum temperature. Optimum temperature means that this is the temperature which the reaction rate is the highest, around 30 degrees. This is the first graph right here. The second graph represents a second enzyme whose optimum reaction rate is reached around 70, 72 degrees. Now, this enzyme was isolated from a heat-resistant bacteria that lives in hot springs. So its enzyme, the organism must function at higher temperature, and its enzymes work at higher temperatures. So up until this point, the enzyme will work great. If you increase it above 72 degrees, however, the enzyme will begin to denature. These are two different proteins, two different enzymes. They have two different optimum temperatures at which they function. That means the two enzymes are able to maintain their shape at different temperatures because they're isolated from different organisms. This is a human enzyme. This is an enzyme isolated from a thermophilic bacteria. Now, similarly, if you look at the lower graph, there's two enzymes that have different optimum pHs. Again, optimum means the pH at which the reaction rate is highest. Those are the best conditions for this enzyme to work. One has an optimum pH of about a little less than 2, pepsin, and the other one, trypsin on the right, has an optimum pH of around 7.5. The first enzyme was isolated from our stomach. Stomachs are highly acidic to help us digest food. So it functions optimally at a very low pH. The second enzyme is isolated from the intestines, where the pH is around 7.5. So the two proteins are very different. They have different shapes. They maintain their shapes under different conditions. Now, my challenge questions to you is, why are the pH curve narrower bell curves than the temperature curves? And the answer will be given maybe sometime next time. Right now, I don't want to give you the answer. I want you to think about it. They're bell-shaped curve because we're approaching optimum conditions, and past optimum conditions, the reaction rate falls again. But why is this bell wider than this one? This is temperature and this is pH. And my hint to you is look at the scale of the x-axis. Okay, so enzymes can be regulated. Enzymes can be activated and they can be inhibited. And here's some interesting trivia. There is such a thing as irreversible inhibition. That means once an inhibitor binds to an enzyme, that enzyme is dead, permanently dead. It cannot function. Irreversible inhibitions are usually poisons and drugs, heavy metals. This is how a lot of poisons work. They bind to a crucial enzyme, and that enzyme can no longer function. But this is not what we're interested in. We're interested in reversible inhibition. Reversible inhibition and reversible activation, because this is what allows a living organism to regulate its chemical reactions by regulating the activity of its enzymes. Now, inhibition can be either competitive or non-competitive, and another term for non-competitive is allosteric. And I'll explain in a second what that means. But let's go back to poisons, because I love talking about poisons. Here are some examples of irreversible inhibitors. Cyanide is a potent poison that can kill you very quickly. It inhibits an enzyme called cytochrome C oxidase, which is the last step before oxygen in the electron transport chain during cellular respiration. If you block the last enzyme, no ATP is produced, and very quickly a person will die. The antibiotic penicillin is another poison, except it poisons bacteria. It binds to the active site of the enzyme, which generates the peptoglycan wall of gram-positive bacteria. So the enzyme will actually change the enzyme, the antibiotic penicillin will actually change the enzyme and effectively destroys the bacteria's cell wall. Bacteria divide every 20 minutes. If you can't make any more cell wall, bacteria cannot divide. So this is how penicillin, the antibiotic penicillin, works. It inhibits the enzyme responsible for making the cell wall of gram-positive bacteria. All right, reversible inhibition. On the picture on the left, you will see the enzyme, its active site, and its substrate. Now, competitive inhibition is when the inhibitor binds to the active site, and when it binds to the active site, it prevents the binding of the substrate. It does not have the exact same shape as the substrate. It mimics it, it's similar, but it's sufficient binding so that it'll prevent the substrate from binding. So they compete. Both substrate and inhibitor compete for binding to the same site. A non-competitive or allosteric inhibition means that the inhibitor will bind elsewhere on the enzyme, not on the active site. But by binding there, it'll change the shape of the active site, and it will reduce binding to the substrate. It will reduce the activity of the enzyme. So it binds elsewhere, but it affects the active site. It affects the binding to the substrate. It affects the activity of the enzyme. Now, how can you reverse competitive inhibition? Simple. You add a lot more substrate, okay? So think of it this way. You have 3 chairs in the room. Normally, these chairs are reserved for girls only. And you have 3 girls. So 3 girls, 3 chairs. The 3 girls can sit on those 3 chairs. You have 1 boy coming into the room who's going to start competing with these 3 girls for the 3 chairs. You have 10 boys in the room. If you have 10 boys in the room, what's the likelihood of a boy sitting in the chair rather than a girl? Very high, because you have a lot of inhibitor, a lot of boys. But if you have 50 girls in the room and 10 boys, the likelihood of a girl sitting on the chair is higher. So you can reverse competitive inhibition by having a lot of more substrate than inhibitor. Now, here's a great analogy for how competitive and non-competitive inhibitors work. On the left, you will see these plastic caps that people that have small children in the house use. They plug them into electrical outlets so that kids cannot stick their fingers in their forks or anything metal into the outlet and get electrocuted, right? Because little kids, toddlers, they crawl everywhere and they want to put everything in their mouths, and they put stuff into electrical outlets. So for safety reasons, parents would put these plastic caps on top of electrical outlets so their small children cannot access these electrical outlets. So once you put a plastic cap on this, you cannot plug in a regular plug because it's blocked. So the plastic cap functions as a competitive inhibitor. Now here's an example of a non-competitive inhibitor. Here's the regular plug where the substrate is supposed to bind. And right next to it, there is another plug where you bind something, you plug in something else. But once you plug in that thing else, the second plug cannot really fit very well. See, it just doesn't go in because they are the non-competitive inhibitors in its way. So this is how allosteric sites work. The inhibitor binds elsewhere. It doesn't bind to the actual active site, but it prevents the substrate from binding very well to its active site, and it prevents the enzyme from functioning. Now, why is enzyme regulation important? Regulating metabolism via allosteric modulation of key enzymes in metabolic pathway is key. It is very, very important. Cells do not want to waste resources. Cells do not want to waste energy. So they would pick a few key enzymes in a metabolic pathway to regulate. Allosteric regulation may either inhibit, that is negative regulation, or stimulate, activate, that is positive regulation and enzyme activity. And allosteric regulation occurs when a regulatory molecule binds to a protein at one site and affects the protein's function at another site. Again, non-competitive or allosteric are similar terms. And we have seen something very similar when we talked about feedback regulation. This is a metabolic pathway. Enzyme 1 leads to the production of intermediate A, enzyme 2, enzyme 3, enzyme 4, enzyme 5, and so on. And the end product is right here. We want to make, this is the reactant, and this is the product. It goes through several different steps, several different chemical reactions until you reach the product. But once you have enough product, you don't want to be wasting resources. So the product itself is going to inhibit the first enzyme of the pathway, enzyme 1. So isoleucine, which is the product, will bind to the enzyme that catalyzes the first step in this pathway and inhibit it. But it doesn't bind where the active site is, it binds elsewhere. Regulatory enzymes are usually at key junctions in metabolic pathways. And regulatory enzymes usually have more than one allosteric site. It's not just one, there's usually multiples. And some of them are activating sites, some of them are inhibitory sites, because cells want to be able to fine tune. It's not all or nothing. They can just fine tune the activity of an enzyme. So here's an example, allosteric regulation. This is one of the most famous enzymes that is so often used in biology exam for students. The enzyme is called PFK1, or phosphofructokinase 1. It catalyzes a crucial step in glycolysis. It turns fructose 1-phosphate to, it turns fructose into phosphofructokinase. Okay, it doesn't matter what the compounds are called. It is a crucial step in glycolysis. Now, when the cell needs a lot of energy, this enzyme is activated a lot. When the cell does not need energy, the enzyme will be downregulated, will be inhibited. So ADP is an activator, whereas ATP is an inhibitor. Why? If you have a lot of ADP in the cell, that means you've run out of ATP. So you need to make more ATP. Glycolysis produces ATP. If you have a lot of ADP, you're going to be activating the enzyme. However, if you have a lot of ATP, that means you have enough energy. There's no need to break down glucose. Let's store the glucose for later use. So you're going to be inhibiting the enzyme. The ratio between ADP and ATP is sensed in cells. Why is it sensed? How much energy do I have? What are my energy reserves? If I don't need ATP right now, I'm not going to be producing ATP. I'm going to be storing all that energy in glucose and glycogen in whatever other means cells have. Plants will store it in starch. So the enzyme is either activated or inhibited because it regulates a crucial step in glycolysis. And here's a graph showing you how the reaction proceeds. In blue, you will see the normal reaction rate when you have low ATP. In green, you will see when you have a lot of ATP. When the cell has a lot of ATP, the reaction will still proceed, but much, much, much slower. So the green is when you add a lot of ATP to the reaction mix. The enzyme is inhibited significantly. It's not completely shut down, but it's inhibited. Now, if you mix ATP with some AMP is adenosine monophosphate, that means that AMP is an allosteric activator. You're going to have some kind of intermediate reaction. That is because the cell plays around with the activity of this enzyme depending on how much ATP and how much energetic needs you have. So you may have a lot of ATP, but you might have a high energy demand right now. So you're going to modulate this enzyme. So the enzyme activity, if you look at this, can vary quite a large range in activity. It can go from perfectly high functioning to very low functioning to some kind of intermediate state, depending on the energy needs of the cell. Cooperativity is a form of allosteric regulation that can amplify an enzyme activity. Enzymes that have quaternary structure, they're composed of multiple polypeptides, they're composed of multiple subunits, are often activated cooperatively. So one substrate molecule primes the enzyme to act on additional substrate molecules more readily. And here's the example. Here's the inactive form of the enzyme. Substrate binds to one of the 4 subunits. It stabilizes the active form, and now other substrate molecules can bind. Cooperativity is allosteric because binding by a substrate to one active site affects the catalysis in a different active site. Of course, there are other types of enzyme regulation, and some of them we've already encountered. Compartmentalization in eukaryotic cells. Some enzymes reside in specific organelles, mitochondria, lysosomes. So you're going to regulate a chemical reaction by sequestering the enzyme away from its substrate. So the enzyme is present in the lysosome. And if you don't want the chemical reaction to happen, you just don't add this, you don't transport the substrate into the lysosome. You leave it in the cytosol. But if you want the chemical reaction to happen, you transport that substrate into the lysosome where the enzyme is. So the enzyme is going to sit in the lysosome with no substrate present unless you want the reaction to happen. There are also covalent modification. Remember, we talked about kinase in unit 4. Phosphorylation. Kinases are enzymes that add phosphate groups, and phosphatases are enzymes that remove phosphate groups. That is the most common means of regulating enzyme function. And sometimes enzymes are subject to, brace yourself for this term, proteolytic cleavage. That means part of the protein is removed, it's cleaved, it's cut out. That can activate, usually activates an enzyme. And this is the end, my friends. Hopefully enjoyed the journey.